Then we can move into stem and progenitor cells when we're done talking about cell differentiation or cellular. Then we can move into talking about stem and progenitor cells when we're done talking about the control of the cell cycle and cellular division, because it's important to really focus on, if we're going to talk about cell differentiation, if we're going to talk about controlling cell division, really it should be important to talk about where all of these cells are ultimately coming from. And an important thing for us to recognize is the idea of stem cells and stem cells versus progenitor cells, and how they kind of differentiate or differ from one another. So when we talk about stem cells, what we're really talking about is that like original, right? The stem cell is really the original cell that everything else starts out as, and I mean, it's not just one, there are multiple, but these are unprogrammed cells. They don't have any specific designation. They're not going to differentiate to become a specific specialized cell. None of that has been decided yet for stem cells. Versus progenitor cells, we refer to them as being committed cells because their relative destination has kind of already been decided, at least to an extent. We can also talk about stem cells being self-renewing, and that's the idea that these stem cells can divide into other stem cells. So when they themselves divide, they can create more stem cells, which is a different feature for many other cells. So they can also divide to create one stem cell and one progenitor cell. And in that case, obviously the progenitor cell already has its destination figured out and it's going to head in a different direction, while the stem cell can once again divide and become two stem cells or a stem cell and a progenitor cell. So we refer to the idea of stem cells being totipotent or pluripotent, while progenitor cells we only use the word pluripotent. And what totipotent means, we're really talking about total potential. This is the idea that they can become any type of cell. They have full potential, total potential to become any type of cell. Nothing has been determined, and nothing has been programmed. So they are, basically a complete blank slate, totally unprogrammed. While pluripotent refers to the fact that they can only become select types of cells. And what I mean by that is, by pluripotent, their specialization has been somewhat determined. This is the idea of stem cells later through the progression, later throughout division, and progenitor cells. They're going to be pluripotent because they're not going to have the potential to become any specific type of cell. They're going to kind of have a specific class or a specific group of types of cells that they're going to belong to, and then they'll be able to become anything within that group. So there's kind of limitations set for pluripotency. So this picture here is just kind of showing the idea that a sperm cell and egg cell, once fertilization takes place, we're going to have that initial zygote, that fertilized egg, and it's going to divide to create two stem cells. This is the very initial stem cell, two stem cells, and those stem cells can divide to create more cells, like it shows here, or they can divide and create progenitor cells. And those progenitor cells are going to be a situation where they're kind of already headed in a specific direction. So here we have a progenitor cell that's moving in a direction for the epidermis, or for the integumentary system, sebaceous gland cells versus actually epidermal skin cells. Well, in this case, the progenitor cell's destination, programmed decision, is going to be into the nervous system, where it's going to become neurons or astrocytes, so on. Neuroglial cells, whatever. Down here we have progenitor cells, and they're moving towards blood cells. They're going to become blood cells, platelet platelets, sorry, red blood cells, white blood cells, and become a part of the blood, the circulatory system. While as our progenitor cell here may become connective tissue versus bone cells, they're kind of going to be stemming off the same initial progenitor cell because they're all kind of similar in their structure and function. Likewise with the blood cells coming from the same progenitor cell that's kind of differentiating out as it divides. So that's kind of that idea of cell differentiation and specialization that we touched upon in earlier lessons, which leads us into a bit more explanation about cell differentiation and the idea that this is how they develop with different structures and specialized functions. And as you saw in the previous picture, it starts pretty early on, and early with divisions right after fertilization for cells to really start differentiating and to start obtaining their specialization. And it's really a reflection of genetic control. And what I mean by that is that basically all cells, except red blood cells have no nucleus, right? All cells have the same genetic info. So all cells have same genetic info. I'm meaning within one organism. So all of your cells have the same genetic info, except red blood cells, because red blood cells have no nucleus, and no nucleus means no genetic information, means no chromatin. But everything else has the same genetic info. Some cells use certain genes. Other cells use other genes. And what I mean by that is that reflecting genetic control, that depending on which specific genes a cell is going to utilize and which specific set of genes a cell is going to ignore, that cell is going to develop certain characteristics, certain shapes, certain structure, and pretty much certain functions in order to meet those structures based on the genetic information that it's using. So the idea that smooth muscle cells are going to be using different genetic information than a nerve cell because their requirements, their needs are going to be substantially different. Likewise, so are their actual structures within based on the genetic information that they actually utilize in order to develop such different cells. So this is really just showing us a simple idea of cell differentiation, which we saw in the progenitor and stem cell image as well, and the idea that how everything can kind of span out from one place, and as divisions reoccur, the cell becomes more specialized. Then we have cell death, the last thing that we're really going to talk about, apoptosis. The idea that this is programmed cell death. What I mean by programmed, it's deliberate. It's something that your body is actually in control of and is doing deliberately as a protective mechanism, as a way to prolong the life of the organism, your life. And it's not something that's accidental, damaging, or anything. And this is a continuous process that's always going on to make sure that the organism you stays healthy. And it occurs at a certain checkpoint right after G1, where after G1 the cell grows and a decision is kind of made at that point where is the cell supposed to proceed through division, or is it supposed to be destroyed because of the potential harm it could cause to the organism as a whole. A really good example of this is how when you get sunburn, you damage the skin cells. You don't kill them, but you damage them. As a protective mechanism, your body actually kills those cells and actually destroys them itself, apoptosis. The cells actually reach a checkpoint, recognize that they're damaged, and that damage is potentially dangerous, potentially cancerous. So the cells die, apoptosis occurs, and you wind up with skin cell peeling. So your sunburn, the skin starts to peel away. And that's really because your cells have essentially killed themselves in order to prevent cancer from developing, because they were damaged cells that could potentially become cancer, and your body recognized that possible flaw and wanted to prolong the safety of you and wanted to ensure proper health and functioning. So apoptosis, we're talking about a deliberate, programmed cell death that occurs as a part of a plan to kind of improve the overall health. Guys, this pretty much wraps it up for our overall lesson on cellular division. If you guys have any questions, please feel free to see me.